So the title of my talk is How Will We Find a Cure? And obviously the disclaimer is that finding a cure for any kind of disease is a monumental task, and very often we have to be very realistic about it, whether we can find any cure. But before we achieve this goal, there are many different steps which we can take to improve lives of patients and understand the disease and help. So obviously, as I said, we will be talking about research. So a quote from our favorite Albert Einstein: if we knew what we are doing, it wouldn't be called research. So in many cases, when I talk or when you read something about dystonia research, you say, well, this is really strange. Why are they doing this? Why are they investing time in something so elusive and esoteric? And sometimes it takes time and a lot of know-how to understand why certain things are done, and they may hold promise for finding a cure or finding a treatment, and it takes a lot of technical knowledge to make those connections. And I will try to simplify a few things for you. So although we are doing research in general as a dystonia field, so we are by nature very chaotic and we are trying to find things out, we nevertheless have some directions, and DMRF is very diligently tries to define those directions. So we definitely know that in dystonia we have to pay attention to those general directions. Genetics of dystonia, we have to concentrate on proteins which are involved in dystonia. We have to find common mechanisms which govern all different forms of dystonia and so-called neighboring diseases like Parkinson's or Huntington's. We heavily invest in to understand neurophysiology of dystonia and essentially neurophysiology of the human brain. We invest in animal studies because there are many drastic experiments which neither of you would like to accept as something to be done on you. And obviously clinical research is of fundamental importance, that the moment we understand something in animals, in lab experiments, we really want to translate this into clinical research. So the things which we are really obsessed with to understand the mechanisms, and again we are at this point stepping slightly away from human beings, that we have to understand brain function, we have to understand how cells and mostly neurons work, and what those tiny molecules in those cells really do. And human genetics tells us that these molecules and specific cells are extremely important because essentially, if they are faulty, they cause your disease. Some people believe that brain function, something which controls you and controls your movement, that the brain is really a set of tiny engines, and these engines are built of proteins. And the way to improve brain function would be really to directly either fix some of those engines, improve the function of those proteins. Some others say, well, this is not exactly true, because it's true that you are built of proteins and cells, but what is really important is functionality. It's really the electrical function of the brain, which would tell us that this is more like a computer. So forget those proteins, let's address directly the electric activity of the brain. And I'll show you later that both approaches are probably true in some way. Obviously, others think that this is all nonsense, that this is much simpler, and we can find solutions much easier. We tend to disagree with these people. obviously the ultimate goal for us, not the ultimate goal of research, but ultimate goal for us in the dystonia field is really to find treatments, to do something, intervene in your brain, with your brain, intervene somewhere inside somehow, so you can improve your motor skills and other characteristics of the disease. We always have to remember that we are talking about dystonia. So we, although we are doing research on a larger scale, we have to stay focused on dystonia. And this is something you probably have seen somewhere. And DMRF participated in really defining dystonia for the neurological community, that this is definitely a movement disorder. It's definitely a neurologic disease. So if someone tells you it's a muscle disorder or something else, it's not true. It involves muscle contractions. It involves movements, postures, especially when you undertake voluntary action and so forth. But despite the fact that we have this in the name, Dystonia Medical Research Foundation, that we all are talking about dystonia, that I bring up this slide with dystonia definition, we have to keep our eyes open on what is happening around us in different diseases, in general, in neuroscience, because there are many aspects of all neurologic diseases which require fuller understanding of the brain function. This is especially true in dystonia. So we really want to understand common mechanisms. And why I emphasize the word common mechanisms, because there are many forms of dystonia, either genetic or non-genetic, and by neurologic definition, they have to be classified differently, even for trivial insurance purposes. But they must have some common mechanisms. That's why all of you are in the same room, that we are all talking about very common symptoms. So we must have, some common mechanisms. This view has been very unpopular for many years among neurologists that who like to classify and overclassify dystonia. Nevertheless, current research strongly indicates that there are some common mechanisms, and we should try to find out, what they are. And obviously, this comes down again to something you know probably very well about neural function. And here is just a highlight of a synapse, of a connection between two neurons, where the information is being transmitted, and what is involved is a set of proteins, mostly proteins called neuroreceptors. And the way the communication is done, it's not only pure sparks of electricity, it's actually transport of neurotransmitters. So these are the fundamentals which are critical for understanding any neurologic disease, and they are focus of many studies in dystonia, as I will try to show later. So don't be overwhelmed by the complexity of this picture. It comes from a review of Neurophysiology of DYT1 Dystonia, and is actually a result of a study done not on humans but on mice, which carry a pathogenic DYT1 gene. And what was shown is really that there is a tremendous difference, without going into technical details. You see the difference here in reactivity of those neurons. So if you have dystonia, the reactivity of neurons in the striatum, in the basal ganglia, where your motor control is really, your movement is really controlled, is fundamentally different. What are the consequences of this finding? First of all, there is a strong indication that there are very specific receptors which are involved, that there are very specific neurotransmitters which are also involved. This implies that one can use pharmacological treatments which would help dystonic patients to control their movements. And this is not completely coming independently out of animal studies. You as patients know that you can take certain drugs, and these drugs act specifically or non-specifically, I would say, on some of those receptors. So the challenge now is to combine this knowledge from the clinic, from patients, and knowledge from animal studies, and work on pharmacological treatments which would be much more specific, which would get rid of side effects and would target very specific receptors. But obviously for that, we have to understand which receptors are involved. This is a brand new, published just probably 10 days ago, review and hypothesis in a leading neuroscience journal, coming from the same group which produced those results on the previous slide. And they go much farther. They demonstrate that what they observe in animals, in isolated cells, can be somehow transposed or translated into what the brain does. And you probably recognize that in the middle, we have highlighted those basal ganglia structures which control our movement. And very schematically, they show that they claim that all hyperkinetic disorders, and dystonia belongs to hyperkinetic disorders like Parkinson's or Huntington's, where movement is compromised, they all have very similar characteristic. That synapses, those connections between different neurons, are compromised and they cannot get back to normal functioning. This has profound implications on potential therapies, and you already heard about, obviously, DBS and non-invasive treatment or stimulation of the brain. So again, this is almost like justifying existing treatment, but providing a very measurable features in neurons, and again combining this, what I showed you on the previous slide, with an array of receptors and proteins which are involved in regulation of striatal function. Obviously, we have to move at a higher level as well, not only concentrate on molecules and proteins and something extremely minute, which we can only visualize with an electron microscope, but we can also take a patient and do an imaging study. And what it shows, that if you have in this case, just an example, an attack of food dystonia, it's not necessarily that your basal ganglia are lightening up. There's obviously motor cortex, something right in the middle of your top of your head that also lights up, but also cerebellum. And this, again, for years, it was considered an error, a mistake done during imaging and not really well accepted. Last 5 years witnessed really a total breakthrough in this area, and cerebellum is really involved in dystonia as well. And this was proven not only in imaging studies, but also using animal studies and genetic studies as well. So you see that we have to keep an open mind on what's coming up and coming out. Staying with imaging, this morning you heard about DBS. And this is not really to analyze the entire slide, but just show you this right bottom corner here. This is taken from a real patient, so-called tractography, which is done using a sophisticated MRI technique, which really shows how your tracts of your nerves point to specific areas involved in this case, in movement. And why this is done, it's not purely academic. Once you know precise location of those tracts, you can guide DBS electrodes to those places. So this is, again, quite a different technique, which has been enabled by imaging techniques. So again, brilliant physicists came up with those ideas, and this was adopted very quickly by medical community. And now, instead of relying on pure anatomy, simply that I know that this area, the GPI or STN or basal ganglia are right in this place, you can use a functional test and guide those electrodes much better. So to confuse you even more, we are jumping back into the microscopic world, and this is actually sub-microscopic world. This is something which appeared in press a week ago, and comes from MIT, from the lab of Thomas Schwartz, who incidentally has been funded by DMRF for several years and is currently funded as well. And this is an atomic structure presented for the first time to the entire scientific community of torsin A, and compared to torsin A mutant, which causes DYT1 dystonia. And again, it's a very technical schematic showing a very precise atomic structure of those proteins. What is really important to understand that this E303, this is one amino acid which is missing in DYT1 torsin A. So what's the meaning of it? It's not that it's a nice picture and nice atomic detail, but this would allow chemists to design and test drugs which might be interfering with this faulty protein, or maybe trying to fix this protein in some way. Again, it's a very arduous and very long process, but this is the first step, and this is one of the greatest achievements since the discovery of DYT1 gene in 1997. Jumping one level higher, and this is a microscopic level, where you can actually visualize, and again, it's a schematic, to visualize torsin A. We know that torsin A moves around the cell, and this is part of the cell. Again, without going into details, torsin A has many friends and avoids certain people within the cell. And it's fascinating that the last 5-7 years, in the last 5-7 years, we discovered several different proteins involved in torsin A action, and a couple of them actually revealed the function of potential function of torsin A, and also point to a potential way of treating DYT1 dystonia with drugs which would interfere with this process. This is not torsin A, this is not a structure. This is actually a kind of a generic picture showing you how proteins interact with each dot is a protein. So imagine that this red cluster right in the middle is your family. So you are tightly bonded, and you know each other. You know your grandparents, maybe uncles and distant cousins, and then your neighbors and your village or your city, and maybe the entire country. If we counted, this would be thousands and thousands, if not millions, of people, or in this case proteins, and interactions. So it is really fundamental to understand with whom you get in touch with, and this would help people who develop drugs or develop therapies to avoid certain interactions or encourage certain interactions. So to give you a certain metaphor, why do we need this? This is a street map of Paris, and if I told you, go from Saint-Denis to Versailles, and you would say, well, maybe some of you know how to go about Paris, but it would take quite a while, and you would have to find different streets and negotiate this, and then you would hit the river, and you wouldn't know. And it would be very difficult to save very quickly without GPS, obviously. So what we are striving to do is to develop this kind of map for dystonia, not only for torsin A, but for all proteins involved in different forms of dystonia, that we identify those proteins. So if I tell you go from Gare du Nord to Place des Invalides and then turn to this street, immediately it becomes simpler. It's almost like saying, you go to McDonald's and then there's Joe's house, and it's much easier than drawing a GPS coordinates. And the moment we start understanding this street map and we become kind of happy, we see all the buildings, we see all those proteins, something like that comes out. This actually came out right now, a few days ago. And this is a real breakthrough in torsin A study. Don't read the middle of it. It's simply, it comes from Rose Goodchild's lab. Rose is a member of our Medical and Scientific Advisory Council, former grantee. She currently works in Belgium. And her lab discovered that torsin A has a fundamental role in regulating lipid metabolism. And lipids it's a completely different story than with proteins. So if you go out here and do a river walk, you see those big buildings. Let's say those big buildings are big proteins, torsin A and other proteins. And what she discovered is that actually what is extremely critical for all those proteins to stand as tall buildings is maybe concrete in the pavement. And these are lipids, and lipids build your membranes, and your membranes determine how your neurons work. So again, without going into detail, this is a schematic representation of lipid metabolism. In normal case, you have a balance. A bit goes into storage, a bit goes into building your membranes, and everything is fine. And it looks like when torsin A interacts with this somehow and compromises this production. So instead of going into energy production, it may go, instead of going into building membranes, it may go too much into energy production and storage. It's essentially building sort of a useless fat. This has fundamental consequences for our thinking about dystonia in general. And what is also critical, it points that, at least in DYT1 case, there is a big component of membrane development when you develop. So when you are a child and when you grow, depends maybe, depending what you eat, what's your general metabolism, you may actually switch to some kind of membrane abnormalities. This is brand new, and we are waiting for other studies really demonstrating what is going to happen. What Rose did, I go back here just to finish, she proved this concept paradoxically in Drosophila, in fruit fly, but also in human cells which are isolated. I know directly from her that she's already moving on into mouse brain and showing that the same concept is true in the mouse brain. So I jumped up and down the different floors of research, going from non-invasive imaging of a human brain to going somewhere very deep in the cell, going to the level of subatomic resolution. And we always have to remember that we have to take care of all the different levels. So not only genetics, not only the consequences of discovering genetics and analyzing proteins, not only talking about receptors and how cells function in the brain. Something we should also remember that once you start combining those thousands and millions of neurons, you will develop paradoxical novel functions. And one of those functions is plasticity, which means that your sets of your neurons, your neuronal networks, will respond in a very specific way to external stimuli or when guiding your movement. And all the paradoxes of plasticity have direct application to dystonia, where we definitely know that plasticity is compromised. Apart from so-called motor manifestation, which are obviously your major concern, there are non-motor consequences of dystonia. And for years, we've been a little bit reluctant to accept them, or maybe we think that they are secondary, maybe they are not so much related. What appears to be really true is that they are part of the whole spectrum, and we should really pay close attention to them. But obviously, you kind of nod and say, research, nice research, whatever. Tell me how to treat dystonia. And you may have seen this slide or something similar. This was constructed by Joe Jankovic, you see, almost 10 years ago. And it's still true. We have major access of treatment, pharmacologic treatment, surgical treatment, non-conventional treatment as well. What we are extremely upset about is really this left panel, where there is a tremendous limitation of pharmacological treatments. We are extremely happy that DBS works, and DBS, it's not only that we are happy with the success. DBS carries a phenomenal message to the research community, even to those who study torsinase at this sub-microscopic level, because it proves a concept. You can almost cure this disease. You can correct movement in a dramatic way, doing something extremely simple conceptually, simply putting electrodes in the brain and stimulating with a very simple pattern of electric stimulation. And you probably recognize these two guys, Salim Benabid, who actually started the DBS in France, and Maylon DeLong, our scientific director, who provided the right targets. And when I talked, showing you different slides, that sometimes you do something and then you find a proof for that, like in the neurophysiological studies. This is exactly the same here, that Benabid did this kind of just playing with the brain, and Maylon actually showed that you are absolutely right, and just move your electrode a little bit to the left, and this will be it, because I saw this in monkeys. I probably won't spend time on that. You probably heard about this. This is now some sort of a revival of this idea that non-invasive brain stimulation really may be very useful. The tremendous damage was done by people who published anecdotal evidence and very contradictory. So people said, well, I read about TMS in one paper and it says it helps. In another paper, case report doesn't help. So people were very reluctant to continue this. But now the technology has improved dramatically, and the way, the scrutiny with which this is done is much better. So this, there's a bit, great hope that this can be really helpful, especially for disorders like dystonia, which are functional disorders. But obviously, I mentioned that we are frustrated with this left panel on treatment of dystonia. So we would like to have new drugs, or maybe improve old drugs. And obviously we have to involve pharmaceutical industry and not only academics. Academics can point into certain direction, but we need pharmaceutical industry. And again, don't read the entire slide, just the bottom of it. This is James Black, the most successful pharmacologist of all time. Propranolol, Tagamet, this is what he discovered, and what was introduced to the world of pharmacology. What, apart from very technical ways how to develop drugs, which we listened to his advice, he said that a project needs a champion. And this is what really speaks to our mission as well at DMRF, that we really, we may not have $7 billion to invest into drug pipeline. We don't have huge labs to accelerate this development, but we can be passionate and we can have conviction and we will be stubborn and relentless. And apparently this is needed. So what next? What next on our tiny, tiny scale at DMRF is that we are funding research. And again, there's no time for me to go through details. You can look this up on our website and see what we are funding. And you immediately, by scanning the titles, you recognize that we fund research on proteins, on torsin A, on receptors, and going into translational stuff. We are very proud of this collection of grants we are funding. We recruited extremely good labs and good people, and are expecting very good results from those projects. We are also happy that NIH continues to support dystonia, although not at the level we would like to have. We would like to have more and more, multiplied by 10 or by 100. But my latest scan, done probably less than a week ago, shows that there's 116 projects which are dystonia or dystonia related, with the direct consequences for dystonia. And the total funding is per year is about 45 million. If we just dissect those which are purely dystonia, this is probably a little bit less, 15 million, and this is what NIH typically reports. But there are new projects which are very relevant to dystonia. So as I said before, we cannot close our eyes and just focus blindly on dystonia. We have to keep our eyes open what's going on in neighboring fields. So apart from research and this sophisticated selection of what should be done and what should be learned and how to interact with pharma companies, we would like to build bridges between people who don't even realize that they could actually talk to each other. So whenever we organize a meeting, this is our motto, this is our driving principle. And I believe that we've been quite successful in bringing to the table people who normally wouldn't really talk to each other, not that they wouldn't like each other, but they were not aware of the particular opportunity or the way they could collaborate. And again, to stay true to this word, our most recent request for applications also required kind of obligatory collaboration. So we really favored people who come collaborating. So almost to end this, I would also propose that DMRF should change its name, or at least we could, instead of saying Dystonia Medical Research Foundation, we could also say, at least from my perspective, because I deal with science, as discovery motivated research funding. And this is what I believe what we do. So going back to the dystonia cure, this is really still up in the sky. It's a long way to go. But as I always say, there's no U-turn. Let's push forward. Let's go forward. So that's all, folks. And if some of you are still awake, I will take some questions. Okay, so the first question is about what is known about DYT25 or genal gene. This is one of the genes which has been discovered relatively recently. Fascinating story, discovered in a kind of an endemic population of Amish Mennonites. And for a while it was thought that this is really a peculiar gene because of very unusual population. Since then, we know that the genal is actually not so uncommon. It has been detected in other families around the world. And the gene codes for a very peculiar protein, which is called G-alpha OLF. And this is a protein, I mentioned membranes, neural membranes. I said something about lipids. This is the gene codes for a protein which sits underneath the membranes and waits for signals coming to the cell, to the neuron. And depending what kind of signal the cell gets, this protein interacts with, talks to the different proteins. And so in a sense, it regulates function downstream in the cell. What was paradoxical in this case is, unlike Torsin A, when it was discovered in 1997, it was totally unknown. That's why it got its name, Torsin A. So the investigators had this privilege to name the protein because it was totally unknown. In this case, when Laurie Azelius and her team discovered the DYT25 gene, they quickly realized, it pops up on your computer screen, that this is a protein which had been known for many years. And in France, there is a guy called Denis Hervé who spent his entire career studying this protein, developing animal models and everything. So in the original paper, they published this together. And they simply capitalize on the knowledge developed over more than 20 years on this gene. This gene is linked very tightly, as I said, to membrane and to receptors. And this opens up a great opportunity to intervene pharmacologically. And if you recall the DMRF-funded grants, one of the grants actually explores the possibility of combining the knowledge of genal and so-called muscarinic receptors to intervene in dystonia. Okay. The next question is
So we only have time for one more question, so I'm going to ask you to be brief.
Yes.
But this is an important question, so I want to make sure that you get to it. How does the DMRF coordinate or collaborate with the NIH? And for those of you who don't know, the NIH is the National Institutes of Health. That's the department that is funded. It's a governmental department that is funded to conduct research on all kinds of things, including dystonia, but everything.
So we've been collaborating with NIH for many years, and primarily with NINDS, which is National Institute for Neurological Disorders and Stroke. Each institute at NIH has a specific portfolio of diseases, and dystonia falls mostly under the wings of NINDS. And the collaboration is very tight. It starts with programmatic issues. We some years ago we had a joint, very big conference where both DMRF and NINDS equally contributed to development of a program for dystonia at NIH. We apply for grants from as DMRF for meeting grants. We are part of, as DMRF, we are real part of the Dystonia Coalition, which is an initiative driven by NIH, where you probably heard about this. It's a massive program to organize everything and anything about dystonia, not only in the US, but around the world. We are constantly in touch with NIH staff, and I'm pleased to say that they consider us as a major entity responsible for dystonia research. So we share information with them about our grants which are funded. The program director from NINDS, who is responsible for dystonia at NIH, regularly comes to our meetings and it's part of the entire process. So it's a very close corroboration. So very quickly, someone asked about, is it true that it costs $1 billion to bring a new drug to fruition? Yes. Some people, Wall Street, they say it's now close to $2 billion, unfortunately.
But the caveat to that is that there's also some drugs which can be repurposed, so they might already be approved for some other disease, but then you find that they have applicability towards dystonia, and then you're not starting over from scratch, so it doesn't cost $2 billion. So there's hope for all those people who don't have $2 billion. All right. So thank you, Jan.
Thank you.